Hey everyone, we're going to begin. This is the fourth and final clinical trials um, workshop, and we're going to continue with Austin Glick, who is the clinical data manager for the Office of Clinical Trial Development and Implementation. And he gave an excellent presentation last week on part one of this. And today he's going to go on to clinical trial data management two. And after him, we're going to have Dr. LaMonte come back, and he's going to be talking about designing pilot trials. So I want to welcome you all to, again, the BERD clinical trial development or clinical trials in general workshop series. And I'm going to turn it over to Austin Glick. Thank you.
Thank you. All right, welcome everybody. So yes, this is Clinical Trial Data Management 2. This is a direct continuation of the presentation that I gave last week on Data Management 1. So this is going to go into a little bit more specifics. We'll show some examples. I'll go through some of the systems that we actually use as data managers. And then towards the ends, we're going to kind of talk about a little bit of the future of the field and what that looks like. So I have two learning objectives for today. First is to apply various data management tools and techniques to effectively manage tasks throughout the life cycle of a clinical trial protocol. And then our second is to learn how to understand and implement key data management concepts within the context of clinical trials. So we're really going to try to apply what I was talking about last week. So how does a data manager take the systems that we use and all of the day-to-day tasks that we need to do, and how do we do them? How do we apply some technology to help us with that? And then how do we go through when we're actually working on a clinical trial? And I want everybody on the call today to kind of think about these concepts as we're walking through. And I'm going to leave a good amount of time at the end for kind of a Q&A session. If anyone has questions about anything that I've talked about, any questions about any of the software that we use, the differences between certain systems, or really importantly, if you have any scenarios where data management is involved that you've encountered in the past, or maybe something that you're planning on encountering in the future, and you have any questions about that, I'd love to hear that and love to let us open up at the end so that we can go through those things and I can kind of walk through what we use and the technology that we have and how to apply that to those situations. So just think about that as we're moving through these slides. All right, so we're going to start by talking about the biggest technological component of data management, and that is electronic case report forms. These are also known as ECRFs. If you remember from the last session that I did, these are how we capture data. They're within an electronic data capture system, or EDC, and these are the forms that you are completing or the data manager at the site level, study coordinator, whoever is keying the data. These are the forms that they are completing and they are putting into the database that is then being mapped to used for statistical analysis. So these are incredibly important. There is tons of documentation and training and huge conferences all over the world that talk about ECRFs solely and specifically. And this is a very, very big portion of data management. So we're going to go through this at the lens of UB, and we're going to look at two EDC systems that are available to University of Buffalo employees, and that is REDCap and Encore. So I'm going to go through both systems. I'll show you what forms look like in each of them, go through some of the limitations and some of the benefits of using either or, and we'll kind of compare and contrast some actual real forms based on CDISC standards towards the end of it. So we'll jump right in here with some of our key bullets for each system. So for REDCap, it's free. That's the biggest thing here. Both of these systems are available at UB. Encore requires a license, which is paid for by the Clinical Research Office. REDCap in and of itself is free. It is a open source software that can be used by any institution, any person. They just need to be able to set it up. It is an easy to use system. Most people who have done some form of research are familiar with REDCap, and if they're not, it's one of the first things that they're exposed to. And it's pretty easy to use. It's pretty easy to pick up. There's tons of things online that you can watch YouTube videos. You can go read a lot of papers on setting up REDCap databases. Tons of information out there. REDCap allows for customizable data collection instruments. These are also the ECRFs that we talk about, and you can create whatever forms that you want. There are some limitations, which we'll go through in a couple of slides, but you can customize things to the most part for what you need. There is real-time data access and management. So this is kind of a staple of an EDC, is that you can go in there in real time, access your data, and manage your database and your ECRFs. So REDCap gives you those capabilities for a cheap, a free utility that you can implement, and it's a really great component. REDCap allows for data export and integration. REDCap actually has a very good export system in it. If you want to pull data from REDCap, you can pull it in a number of different ways and different file types, more importantly. If you want to pull your data from REDCap and analyze it, you can pull it in a SAS file format, just straight from the system, which is great for doing type of reports, or going through if there's a little bit of an analysis, you want to explore some of your data in the middle of a study, or you want to create a custom report because maybe a PI or you're the PI and you want to kind of get an idea of where your study is at. And then REDCap also has a very nice feature called Participant Online Surveys. And this allows you to create an ECRF within REDCap and then create a link that would publish that form for that participant to an online component that you could then send to the participant and have them fill out. So this is a great component of REDCap. It's something that they implemented pretty early on, and it's fantastic for letting participants fill out some surveys. A lot of people I know use it for consenting as well. Like it is viable for online consenting. There's some regulatory components behind that, but it's there and it's capable, and it's a great little utility. Now, if we jump over to Encore, Encore has some, I would say, higher level capabilities, but it also comes with the cost of the license and things like that, and it's a little bit more complicated. So Encore is a centralized cloud-based data management system, and the key kind of phrase there is the cloud-based component of it. When you're using REDCap, if you are doing a study at the University of Buffalo and REDCap is housed within our UB REDCap system, it's fine. You don't have to worry about any cloud-based mechanics. If you're doing a large multi-center study, you have a lot of sites that are outside of the university system, you run into a lot of problems with REDCap because centrally we need to manage REDCap ourselves. Encore, as a company that runs a cloud-based system, anybody can create an Encore account. They have to be invited to a study, and Encore will then manage those individuals who have access to the system. So it's a lot nicer for multi-center studies, having lots of staff at different sites being able to access this. And another important component of that is then Encore, Advara is the company that owns Encore, would take care of dealing with the more help desk information. If somebody loses access to something, then UB is not responsible for necessarily helping them get their access back really quickly. Encore has administrative integration. A lot of people might not be familiar with Encore, but if you are, you're probably familiar with the CTMS system, Clinical Trial Master system, that's in place. That is what university and what the CRO, Clinical Research Office, uses to oversee, any of the tasks when it comes to the administrative components of clinical trials and the financial components of clinical trials. So it does have that integration right in there, and the EDC portion of Encore, it connects directly to that administrative side. So it's really nice on that side. Encore is 21 CFR Part 11 compliant off the bat. If you don't remember what 21 CFR Part 11 is, it is the regulatory component from the NIH that says that all electronic data capture systems need to have an audit trail. They need to follow a lot of guidance for how things are presented and how they're completed and how things are stored. Encore, being owned by a company that solely provides EDC systems to the United States, are compliant with that policy from the get-go. They do not let you do any changes to it that makes it not compliant anymore, because then they would have issues with that. So it is totally compliant out of the box, and it's another thing that you don't have to necessarily worry about. Encore has many advanced reporting tools as well. You can create incredibly customized reports within Encore and make them rerunnable, do whatever you need to do with that, which is great. And that's kind of a big thing within larger EDC systems. Encore also has access to data-driven insights within the main hub portion of Encore. So you can see some insights into your data as you're going through. Once you log in, you can customize the widgets that are available on your homepage. And that is a great little utility. Encore also has the study management tracking and reporting. Kind of talked about that. Those are all just those components of the more administrative side. And then Encore has great integration and interoperability. So it has access to a couple things that I'm going to talk about in the future, like ePro patient reported outcomes, which is like online questionnaires that the participants complete themselves, and eSource, which is connecting to the EHR directly and pulling the data into the database. So these things are available within Encore. Also, a DSMB component is available on Encore as well. So these are really nice type of modern and future features that are great to have. So now we'll jump into some kind of field options and we'll go through REDCap and Encore. And for the next couple of slides, we're going to jump back and forth. I'm going to show you what REDCap looks like and I'm going to show you what Encore looks like. And you'll see kind of the differences, the pros, the cons of each system. So for field options, REDCap has a lot of choices. You can do text box, notes boxes, calculated fields, multiple choice with either a drop down or radio buttons, different just formatting of it, check boxes, yes, no, true, false, a signature field, which is great if you want to ever do consenting through REDCap, through online consenting, a file upload feature, so participants could upload a file if they needed to for anything like that. Slider, visual analog scales, descriptive text, and then just like section headers. There's a lot of field options. REDCap kind of breaks them into more specific fields. If you see, we'll go to the next slide here. Encore kind of keeps it a little bit more broad. They have just basic options: attachment, calculation, date, time, duration, number, options, text, and time. And within those field types, you can then customize them however you want to then make them more specific. So, if you want a yes and no question, you would pick the options field. So in REDCap, you would pick yes, no. That's the basic one. In Encore, you would pick the options field, and then you would create a dictionary, or you might already have a dictionary that's available that has the options yes, no. And it might sound simpler to do REDCap for this, which it is in this situation, but where Encore succeeds in being a little bit more future ready and ready for more complicated studies is REDCap's is yes and no. Encores can be yes, no, and maybe other options if you need it. So if you have a question for a laboratory thing and it was a self-reported by a participant, and you can have yes, no, or unknown, in REDCap, you'd have to then create a custom field to make yes, no, and unknown as individually versus using the yes, no option. Versus in Encore, you can just create a dictionary. You can use the text option or the options option, and then you have yes, no, and unknown. And you can create a bunch of these dictionaries and keep a dictionary repository available that then you can tap into, and you only have to really do it once. So there's really similar options between the two of them. Encore has more customizability, but is also a little bit more complicated to use in the startup. Takes a little bit more getting used to. They both contain all of the recommended field types from CDISC. So that's our data interchange standards, that consortium that they have available. It is the gold standard for how to conduct good clinical data management. They set like how we should create new questions in ECRS, how the language should look, what the options should be, how we should label dictionaries, how all the way down to how we label the fields within forms. So CDISC is kind of the gold standard. Both systems have the ability to create whatever you need for CDISC. Biggest difference between the two is that Encore allows for log forms to be created and REDCap does not. Now, a log form is something I'm going to go into more detail when we start to go through the actual examples, but it is a big component of capturing data, and there's a lot of issues that come for not being able to create logs. So we'll see that in the future. So next, we'll talk about edit checks. Now, edit checks, for those of you who aren't aware, also referred to as branching logic. This is how you create a form that you differentiate an online form. Okay. You differentiate an online form from that of a paper form. In a paper form, you hand it to a participant. They see all the questions. They could complete all the questions and not follow the instructions. On an online database in an ECRF system, we can create logic that allows us to restrict questions, skip questions, reduce the amount of answers that participants have to, or the sites have to choose from, things like that. And this allows us to make a really intuitive and well-flowing form. Um, so in REDCap, the options that we have are right here. There's two big options. It is typing out your branching logic using smart variables and special functions. You kind of have to code it yourself. And then this drag and drop logic builder, which is a really nice feature. And it makes it easy to create kind of simple edit checks. You know, if you want to pull something and say, you know, if a question is asked on the top of a lab form, was the laboratory blood drawn? And you say no, and then you just make the rest of the form not show up. And you could do that in this drag and drop field, which is really great, but it doesn't really work for more complicated things. Um, and the functions are well defined, and there's plenty of help documentation to assist with the syntax when writing within REDCap. You can find tons of stuff online. It's really easy. There's tons of help that you can get to get pretty in depth with some of the edit checks that you could create. Encore, in contrast, has a little bit of a simpler UI than REDCap. You see here, it just says skip pattern, option filter, validation. You can create a label for your actual skips. So you can label them however you want, create your own system, and then go back to those as well and use those in the future, which is really nice if you're reusing forms, things like that. Um, and like REDCap, the syntax needs to be defined manually. And you can see at the bottom of the constraints for Encore, it does have the effect that you want to do, and they break that down into mandatory, deferred mandatory, not applicable, and hidden. So there is a lot of syntax that needs to be written into this one, but you can kind of select two different filters to say what you want to do. So say you want to do an option filter, you would label it whatever you want to label it. You put in your expressions. So say if question A was answered as yes, then for question B, I want to make these items from the dictionary, which you would put in the targets caption, hidden. So then if somebody's in option A hits yes, those options in question B are no longer there. So they can only choose the specific ones that you let them choose for that based on how that form is supposed to be flowing. So it's really nice. It's a simple UI, but it's got a very high ceiling. You kind of have to learn a lot about it. And there's a little bit less documentation for Encore. Most of it is through their learning system and learning management system. So you kind of have to navigate their own trainings. All right, so now we're going to take a look at some specific forms and some examples of what forms look like. So my apologies, some of these might look a little small. I'm going to kind of walk through them, but this is what the forms would look like. So for the first example here, this is a vital signs form. Very common form used in a lot of studies if you have patients in a clinic. So, you know, you start with a word vital signs performed leading question, and this is where you can put some of those edit checks in there. So if they say no, you can make everything else disappear. Maybe you keep date if you want to know when they were in clinic and they just didn't perform vital signs. There is a date field, and then you jump into the results field. So you can have systolic blood pressure, and then the units in REDCap are separate. So they kind of put them into separate fields past the original field. That's going to be a little bit different in Encore. Blood pressure position, location, there's more things at the bottom of this form. I didn't pull it all in. The calendar in REDCap is available for dates. So if you click on that field, you can hit today, you can click the calendar icon, you can select a specific date from the calendar. Very nice. And you can also type in it based on the format. You can change the format of the date as well, depending on if you're using international standards, local standards, whatever you want to do. Unknown dates are not allowed in REDCap. So that can be a problem for certain things. Certain studies, you kind of want to have unknown dates. If you have a medication log and a participant started the medication 20 years ago, they probably don't remember what day they started it 20 years ago. So it becomes a little bit more challenging to key that in the standard version of REDCap. And then the units are set in these separate fields. So now if we jump over, this is what it looks in Encore. I personally think Encore is a little bit cleaner. It's a lot of white, which can be a little bit annoying. So you can move things around a little bit with how you organize it and how you set things up. But it's the same questions. And these forms, I should preface it, these are all the CDISC standard forms. So for REDCap, I pulled them directly from the CDISC standard REDCap library that does exist. You can look it up. You can pull all of CDISC appropriate forms that they've made into REDCap directly. It's great. It's a wonderful utility, and they're all going to be perfect per CDISC protocols. Encore doesn't have that, so I had to create the form myself, but I've been creating this library for our office. And it's not that hard. It's just following the standards set by CDISC. So same questions on here. We have like the radio buttons for these questions in Encore. And it's the same thing. The logic is in there. We can hit, if we hit no, then the other questions will disappear. Encore skipped questions. The question text still remains. The answer option disappears. So that's a little bit different. So the question text is still there. It's not something I personally love. Because people will still see the question text, and it might confuse people when they see it, but they can't answer it. But that is like that. Same thing as within REDCap calendar, you can open up the calendar and you can enter the dates in there. Unknown dates are allowed within Encore, which is really nice. Unknown date and month are allowed. So if the participant, like I said, ConMeds, they only know the year, they could just put the year in. And then units are attached to the field. So as you can see in like systolic and diastolic blood pressure, instead of it being the next field down, it's just it's right there. The unit is right next to the field, which is a little bit sleeker. So then we move on to adverse events. So this is where we talk about log forms. So what do I mean by a log form? In this form right here, this is an adverse events form for REDCap, and it is only allowed to be filled out once. It is a form. You can only fill it out once. In order to fill out more adverse events, you would need to add additional forms. And that can be a problem because participants on clinical trials can have multiple adverse events at any given time. They can be hospitalized for something, and they can have a bunch of different adverse events with that. They can have an injection site reaction, and they can have a lot of injection site different reactions to that injection. And it becomes a little bit more complicated to report all of those AEs when it is a single form for each thing. So there's a couple ways to deal with this. And the two most common solutions are a manual add form button to roll out an AE log. So you put some type of button, some type of drop down, something in the build that lets the person entering the data just add as many AE forms as they want. They have to do that, and they have to follow through with that process. And then they have to also not add too many or go too above and beyond. So there's a little bit more quality issues that come with that. And then the other option is to create the form at every visit and then add a question asking if any AEs have occurred at the beginning of the form. So generally, best practice is to have a log folder called participant logs. And that is capturing all of the things that might be addressed over the life cycle of the study. So adverse events, concomitant medications, things like that, which are going to be medical history, anything that's going to be potentially changing over the life cycle of the study that isn't just at one associated visit. To go away with that process and put an AE log at the start of every visit, and then put a leading question that says, were there any AEs at this visit? And then they can select no, and it just makes the rest of the form go away. And then also add a question at the end of the form saying, were there additional AEs reported? And if they select yes, roll out another form, and you can continue that chain. So those are just some solutions that people have for it. Now, the ideal situation, and this one's a little bit small, but I'll like walk you through it, is that we have something called a log form. And in this situation, in this form, what you're seeing is the top portion, those are all of the questions. And then that bottom portion down there, it is creating a log as you complete the form. So this form, unlike any other normal forms, is whenever you complete it, you're just adding to a running log of completed forms in it, so you can continue to manage this form throughout the study. So in this case, we have AEs. We have the first AE down there, and then I started filling out a second AE on here called AE number 2 for hypertension. So once we filled this out, we would apply it, and then we would have AEs 1 and AEs 2 on the same form. And this lets that form live in that participant logs folder and just stay there forever. And then people can go in and out of that whenever they please and make any updates that they need to. So this is how you kind of get around that. And this is the best process to do things like adverse events. Other one is con-meds or concomitant medications. For those of you who don't know, it's basically any medication that a participant on a clinical trial is taking that isn't the study drug. So just like AEs, con-meds may be collected through the life cycle of a participant on the study. So if they start a new drug, they go to their cardiologist and they get prescribed aspirin, they need to mark that on their con-meds log, and we don't know when that will occur. It's probably going to occur in between visits, actually. So we need to have a log that allows us to capture all of that data in a running manner. So no long forward for REDCap means that we need to do the same thing with AEs, either manually or rolled out at every visit. In contrast, in the Encore form, we can capture everything on one log. We let all the top questions be there, and then we can go through it. And in Encore as well, there's like a little edit button in the log that you can go back and edit anything you want to too. So we can also track that. And it helps us follow the flow of a participant a little bit easier, especially if they have a lot of AEs. So that is kind of the dilemma with log forms. So those are what some ECRFs look like. They can differ greatly. Those were some of the 3 most common forms that you'll see, but ECRFs are based on the study. You can have the most unique form in the world because the study requires one. You can have a lot of questionnaires. Things change and things are different. So picking the right system to use when you start up a study is very important. And now, speaking with the systems, we're going to move on to some utilities. Now, these are things that would be used alongside the system. They're not necessarily required. Not every study is going to use any of these systems or all of them or whatever. But they're things that might be useful on a protocol. So we'll start with randomization. Greg gave a really good talk about randomization last week, I believe. So this is kind of, I'm not going to talk too much on it, just one slide, but I want to say that randomization systems, a lot of them are integrated directly into the EDC system, which is great for us because the data managers have to deal with randomization quite a bit. They're going to put the participant on different arms. It's going to change how the flow of their data works, what forms that participant's supposed to be getting, what they're doing at each visit. Maybe some visits will be not applicable based on the arm a participant is randomized to. So it's really important, and a lot of systems do integrate those directly in. Encore does have a directly integrated randomization system. And REDCap has the capabilities to do a directly integrated randomization system, but you kind of have to implement it through other means. If the system isn't integrated into the EDC, an RN assignment field or form needs to be incorporated into the study build. By that, I mean, when a participant starts on a study, if you're randomizing them elsewhere, you need to somehow tell the system that they're on that arm. So if they're on the control arm, maybe they don't have those safety follow-up visits that are in like the experimental arm. And we need to make it so that the system doesn't roll those visits out so that we're not having people accidentally completing them. Customizability and complexity vary based on the system. Many systems rely on a randomization file. I just put this strat text file in here. This is pretty simple. It's an example from one of the forms and one of the systems. This one's just stratifying for males and females. So this is something that you might be putting in there. And a lot of the statistical side, it comes from the statistician, but for data managers, we're implementing a lot of it. Limits, arm assignments, distribution, and other factors can be updated based on the shifting needs of the protocol. We can change the randomization system as the study goes on. So if we need to shift things, that's something that we can do. So that's something to always keep in mind as we're maintaining the system. And it is essential to properly document any changes to the randomization system or individual participant randomization. Change everything. All the regulatory components say we need to document it and we need to document it well. If we ever get audited, that always comes up. And why did we make any changes? Did we document it properly? So it's always something to consider. So the next one's a little bit newer. It's electronic patient reported outcomes, or ePro, sometimes referred to as electronic clinical outcomes assessments, or eCOA. Data management loves all these acronyms for whatever reason. We live with them and we deal with them. So these are essentially the same thing. They have some key differences, but a lot of the companies advertise them as the same. And this allows the participant to complete questionnaires and some assessments on electronic devices, things like phones, tablets, laptops, could be like an iPad in a clinic. It could be on their individual cell phone if they want to complete things at home, stuff like that. These are directly linked to the EDC. So this data funnels right into the EDC, which is great. It allows us to capture data from the participant directly, which is not something we've been able to do in the past. And then it's effective at collecting blinded questionnaires from participants while they are in the clinic. Historically, if a participant was having a blinded questionnaire, something sensitive, maybe it's drug and alcohol use, health history, things like that, things that are more protected, it was always good practice to blind the site staff from those questionnaire answers. But the standard of doing that previously was to hand them a piece of paper, make them fill out the questionnaire, put it into a sealed envelope, seal it, and ship it directly. And things have been lost in mail. Sometimes those questionnaires aren't filled out properly. Participants don't write their study ID on them or accidentally write their actual name on them, which is unblinding the data manager. So that can be a big problem. And the new way that the field is going is to using iPads with ePro that lets the participant fill out that questionnaire right there on the iPad, goes directly to the database. There's a little bit more guidance on it, and it's perfect. And as you see from this image here, this is just an example of like how cell phone integration will work. This is a company that creates these ePro systems. There's a phone app. A participant can like log pain if it's part of a study that they're doing. And anytime they have pain, they can open up their phone, open up the app, go in there, pick the date that they had the pain, pick where it's located, submit it, and go straight to the EDC, which is fantastic for us. And then the other thing is reports. I talked about reports briefly, so I just want to show you an example. This is an accrual report from a study in Encore in the testing database. It's got 44,000 participants listed on it because it was used to kind of chaos test something. And the report doesn't really make any sense, but I just want to show you kind of how it could output. And it's really customizable in Encore. And this is the general scheme. So when you're creating reports at the beginning of a study, you want to create stuff that could be rerun a lot. And then that gives us the ability to run things like accrual reports on a weekly, bi-weekly, monthly basis and report that to the study team, the PIs, whoever, so that we can monitor the accrual as the study's ongoing. So really great. You can break it down by certain categories like ethnicity, age group, biological sex, things like that, the protocol type, all of those things. So it's really great utility to have. And it's a really important thing to set up in the beginning of a study. All right, so now we'll take just a couple of minutes to talk about what is the future of data management, and then I'll kind of we'll open it up and I'll let you talk about, any questions you might have or any scenarios that you might want to go through. So first, we're going to talk about eSource. This is another big new one. This is automatically populating structured CRFs with the exact protocol required data directly from the electronic health record or electronic medical record, EHR, EMR, whatever it is. Things like Epic, which we'll see in the next slide. It allows you to just pull data directly from their EHR. And this is new. A lot of big EDCs are starting to implement this, and it kind of cuts out the middleman. So instead of having a participant come to clinic, do a study visit, that goes into Epic, and then having a site coordinator or a data manager at the site level take that data from the chart, plug it into the EDC system, and potentially have errors from the reentry, things like that. We're pulling straight from the electronic medical record straight into the EDC. Obviously, there can still be errors in it, but we're doing a direct link rather than with this kind of middle person in the middle that's trying to enter data. So it's great. It enhances data accuracy by reducing manual data entry errors. It streamlines our workflows, making the data collection process more efficient. It's direct. The data is directly entered. Usually it's completed in the EHR immediately when a participant is in clinic. So we're not waiting on that timeline for data to be entered. And then it reduces the time and cost associated with the manual data transcription. So for a site level, there's less cost needed for site personnel to be constantly entering data manually. So it can save some costs there when studies are a little bit more limited. So this is just an example of like Epic's EHR system. It's probably the most common, it's the most prevalent one. We have our patient here, Mickey Mouse, screenshot I could find. And we're going in and we're putting a new problem in there for knee pain. And when we're searching it up, it's pulling up the ICD-9, the ICD-10 codes for that. And that's great. And so when we put that in there and put that in, accidentally hit the button, it's going to go directly into the EDC system, and that's going to mark itself as a adverse event. So we have an adverse event for knee pain now pulled directly from this. There could be issues with it. I mean, there could be things, more information that we need, stuff like that, which we deal with in the future. But for a direct entry, this is great. So then data visual analytics. This is another big one, another big keyword. I'm sure this is the point where we get into a lot more statistical stuff, which everybody on this call probably enjoys. So this is something that is being pulled into data management pretty heavily. And it makes it easier for us to interpret complex data sets. A lot of clinical trial data sets are very complex. It also allows us to facilitate better collaboration among stakeholders. We kind of get a unified platform for data sharing and discussions. These are kind of the big data visual analytics keywords. But when it comes to data management, what we're concerned about is real-time data monitoring and advanced analytics that we can use to to identify issues early, and real-time data insights to assist data managers with QA/QC reviews. So we can take data visual analytics, set it up to a database, give us visual views of the data and what's being done, what's being entered, things like that. And we can use that to affect how we do quality assurance and quality control of the data. We can see, are certain sites entering data late? Are we having a lot of spike of issues in a certain item that we have there? So we can go forward and use that to give us insights at the data management level, and then also share those with the PI and other individuals on the study team. And then these also offer a holistic picture of overall clinical trial progress and supports kind of strong risk-based monitoring and makes it easier to spot areas concerned. Similar to what I said, risk-based approach is kind of the key to modern day data management, and it allows us to use these tools as best as possible. So this is just a simple example. It's a little bit blurry, I'm seeing on the slides here, but it's just like showing AEs for certain data set for a fake study here. And you get a lot of things like how the breakdown of those AEs, stuff like that. This is the kind of things that we can look at and follow and use while managing the data. And then the last one here before we jump into the Q&A is AI and machine learning. So these AI and machine learning algorithms can automatically detect and correct data anomalies, ensuring higher data accuracy and consistency. AI enables continuous monitoring of trial data, allowing for real-time detection of potential issues and timely interventions to ensure patient safety. And these technologies can really significantly speed up data processing and analysis, and reduce the time required to clean and interpret large data sets. So in the terms of data management, this is very, very young. I haven't seen a lot of it going into data management just yet. Some companies are exploring it, and it's kind of pushing the field in that direction. But it is probably where the field is going to go, using AI technology, using machine learning to go through and filter through data. I think, like I said in the last session, we could have clinical trials that have 10,000 participants, and it is just impossible for one to a team of data managers even to review all of that data in real time. So using these type of technologies could really help us review data, find pretty common, simple errors and issues, and address them pretty quickly. I don't know necessarily if it's ever going to get to the point where you can have an AI data manager. I don't think that there is a level of intuition that needs to be there for data management, but it's something to follow and see how it evolves with the field. All right, so that is the end of it. So I wanted to leave some time for Q&A session. It looks like I took a little bit longer than I anticipated.
I think that was perfectly fine. No worries
Okay.
whatsoever. Does anyone have any questions for Austin? He gave a lot of information in a short amount of time, but I think he gave it at a really nice pace. So if anybody has any questions, especially about perhaps your own studies, now would be the time to ask, please. Don't
Yeah.
think we're hearing anything, and there's nothing in the chats. I guess my question is, can they contact you if they
Yes.
need to?
Absolutely. So yeah, if anyone has any questions about any scenarios, any data management stuff, I mean, feel free to unmute now if you want to. But if you ever have any questions, feel free to reach out to me. My office is here to help with clinical trials, and if you have any just like simple data management questions or questions about software or anything like that, definitely do not hesitate to reach out. Be happy to answer them.
Great, and he apparently has helped quite a lot of people upon coming to UB already.
Yes.
So please feel free to reach out because he's always willing to do so, which is wonderful. This was a great presentation, so I appreciate that very
Thank
much.
you.
And I want to tell everyone that Austin's slides are in the chat if you'd like them, and if you would like a recording of this a session along with Dr. LaMonte, we'll be starting in a moment or so. Please feel free to email me. I can put my email in the chat, and I have yours because I know you came after I put it in the chat, so no worries. So please feel free to email me, and I will get you the video next week. And I think, Dr. LaMonte, are you ready?
Yep, I am.
Great.
Can I share?
You can share.
Okay. Uh, Austin, that was a wonderful presentation and very instructional. I have to say, we are very fortunate to have somebody such as Austin with all of his talents and experiences on our UB team. I'm glad you're not playing for the other team.
Thank you. I'm glad to be here. It's been great being at UB and being able to kind of share the what I've learned in industry.
Thank you very much.
Yeah,
I'm going to
thank you.
share now.
Excellent. Thank you again, Austin. We really appreciate it.
Yeah.
And thank you for your flexibility.
Yeah, thank you, and thank you everyone for paying attention to
Thanks.
my presentation.
Okay, Dr. Lamont is coming on with pilot studies. Thank you.
I am. Thank you. Hi, everybody, and thanks for having me back again. Today, I'm going to talk a little bit about pilot studies, and this will be a little bit from where I left off with the introduction to clinical trials, kind of the 500-foot view, which was our first in the workshop installation. And I'm also probably going to pick up on a few elements that Dr. Miller discussed during his time with you. My perspective is that as a whole, the audience doesn't have a tremendous amount of experience with designing and implementing randomized trials and by extension, pilot studies. So that's the point I'm going to be coming from. Now, the life study or the life randomized trial, Catherine, just confirm one more time. You can see this, right? Because
I can see it.
okay,
Not a
I'm
problem.
not my view on my screen is a little funky. It's probably something I've done. Just so you can see it, that's all I care about.
Yes, you probably see your slides and then a bunch of slides on the side. Is that correct?
No,
Okay. No, we can see it.
I think we can roll.
Okay.
So the life randomized trial, which was published, the primary outcomes trial was published in 2014 in JAMA, was a very important seminal randomized trial in the area of aging resiliency. This was the first large-scale trial in the US that showed that exercising older and frail adults can reduce the incidence of mobility disability or basically losing their functional independence. So I'm going to use this trial as a backdrop for the discussion on pilot studies, and you'll see why I'm going to do that momentarily. But just to orient us all to life, the primary trial, the design was targeting frail, elderly, older adults, 70 to 90, The design was a parallel group with one-to-one randomization to the intervention and the control arm. The primary outcome was mobility disability. This was an in-the-clinic test. I'm not going to describe how it was done, but basically, if you didn't pass the test at some point during the trial, you were identified as a loss of functional independence case. Now, what's important for the discussion today? The life trial investigators determined that in order to successfully implement the trial design as designed and test the hypothesis, that they would have to randomize a minimum of 1,600 participants into the two groups, and that by doing so, they would have 80% statistical power to detect a 21% reduction in risk of loss of functional independence. Now remember, that 21% risk reduction would be comparing the intervention arm to the control arm. So that's the I minus C difference. They determined that they would have to do so, run the intervention for a mean of 3 years, and they expected an 8% annual loss to follow-up. Now, the question we need to ask right now is, where the heck did they come up with these numbers? How did they figure all this stuff out and then go on to implement successfully this seminal trial and have the results reported in one of the best general biomedical journals in the world? They did a pilot study, and that's what we're going to focus on today. So that information that they basically used to design and execute the trial was gathered before the trial ever started. And that was in the form of a pilot study. This is one working definition. A couple of key points. It's a small-scale preliminary study. It's done before the actual trial of interest. What's it done for? It's done to determine feasibility. Can we actually do this intervention? Can we actually recruit the people we're trying to recruit and randomize enough of them to groups so that we can test the hypothesis of interest? The pilot study is also there to really work out the kinks in the protocol, test the protocols and refine them, particularly for safety purposes. And importantly, the pilot study is where they came up with the means to estimate the sample size that they would need to conduct the full-scale study. So the pilot study was an integral part of the life trial. What are the typical objectives of a pilot study? Again, I kind of captured this in the previous slide, but just to list them, kind of the more important, not that this is the only thing you would necessarily look to gain from a pilot study, but key things: feasibility, methodology, sample size, sample size, sample size. Can't emphasize that enough. And statistical power. Sample size and power go hand in hand in many regards. So where I emphasize sample size, you can also be thinking in your mind statistical power. Now notice, I said this is done before the trial. This is done to inform developing the trial. One of the things that happens often at the BERD and at the CTSI is when we release our funds for competitive review and awarding of pilot study funds, we get proposals sent in that are essentially the full-scale project. That is not what a pilot study is for. A pilot study is to develop around what you ultimately want to do some key pieces that are going to teach you enough about what you want to do to refine it, to know how many people, how much money is it going to cost me, how long does it have to go, and what's my statistical power for my hypothesis test. Now, the sample size issue is something I'm pretty sure Dr. Miller and possibly both Dr. Miller and Wilding have covered in some way or form. I'm not going to, this isn't a lecture or workshop on sample size and power, but I want you to see how important the pilot study is ultimately for what we really want to know. How many people and how much power will I have? So this is a simple equation to estimate sample size for a binomial test, like a risk ratio or an odds ratio. So we're solving for the required sample size to randomize. There's some key pieces of the equation you should recognize from either Dr. Miller, Dr. Wilding, or both of their lectures. We have to have some idea of what the type 1 error rate is, the alpha level that we're going to test the hypothesis at. Likewise, we have to specify what the type 2 error is going to be, or the beta error. And this is critical because, as you probably know or remember from Miller and Wilding, 1 minus the beta error is actually your statistical power. The other things that we have to know is what is the overall expected probability of the outcome. And then knowing that, we have to have the effect size. What's the difference in probability going to be, or what do we plan on it being between the intervention and the control group? Now, this is nothing more than a numerator term and a denominator term. Question to you. Everything else being held constant, if you make the effect size smaller, what's going to happen to the sample size? The denominator gets smaller. I'm hoping in your mind you're saying, Mike, that's a no-brainer. If all else is constant, if the denominator gets smaller, the sample size has to be bigger. And that would be correct. If everything else is constant and we have an outcome that has a very high probability of occurring, so the numerator gets bigger, what's going to happen to the sample size? All else constant, the sample size would be, or the required sample size would be smaller. And then lastly, if the alpha that we test the hypothesis at is, instead of the kind of conventional 0.05, but instead we decide we're going to test it at 0.01, and everything else stays the same, that means alpha gets smaller. What's going to happen? Sample size is going to go up. So my point in showing you this simple equation is much of what we want to find out from a pilot study, sample size, power, overall event probability or rate, and effect size, all of that is used ultimately to determine the sample size and estimate the power. All of this is done as part of a pilot study. So when we receive pilot protocols or pilot proposals at CTSI, and there's nothing in there about sample size, power, beta error, and alpha error, that tells us the person submitting the pilot study proposal does not understand what the pilot study process is for. And then that proposal gets triaged to a weaker proposal when we rank them for funding. So back to LIFE and the LIFE pilot study. Again, the primary trial was published in 2014, and I will come back to this before all said and done. What I want to focus on is something that was done almost 10 years earlier, and that was the pilot study, which was published in 2005. This is a snip and paste from the LIFE trial pilot study design and methods paper. In this paper, the investigators list 7 objectives that this pilot study was going to accomplish. Pause for a minute. This is a major parallel group design, randomized controlled efficacy trial. Okay. The primary trial is to do one thing: test the hypothesis that the intervention is better than the control condition. Notice in the pilot study, there's 7 things listed that they wanted to accomplish so that they could design the primary trial. Let's look at what those things were. And I want you to keep in mind, this is truly a pilot study. First thing they wanted to do was obtain data so that they can estimate the sample size they would need for the full-scale trial. And to do so, they wanted to come up with an incidence rate or probability that the outcome of interest would occur. Now, a lot of pilot studies do not necessarily involve measurement of the primary outcome in the main trial. A lot of the primary outcomes for main trials, at least in those that I work on, is death or a clinical event like a heart attack or a stroke or Alzheimer's disease or hypertension. So that may not always be relevant or feasible to have as part of the pilot study. But in this pilot study, the primary trial was so unique. It was a high-risk trial. It was working with frail, elderly adults in the 80-plus age range, where there would be a lot of morbidity to begin with. And in order to understand the minimum sample size needed to test their hypothesis, they decided we actually have to measure the outcome rate as part of the pilot study. And you can see here what they define that is: mobility disability, which is loss of functional independence, or death. The or death part, I think, is obligatory because of the age and health status of who they were ultimately trying to enroll in the life trial. The second thing, examine the consistency of the physical activity intervention, the exercise intervention, based on different definitions. So they were going to look at their protocol for the intervention. Third, they were going to try to understand participants' ability to adhere to the intervention and to retain them in the group randomized. This is very important in pilot studies. One of the things we know when we do interventions on whole human subjects is that there's often going to be some level of non-compliance or non-adherence, and also complete loss. They drop out, we have to remove them, and while they started, they don't finish the trial. So trying to have an understanding of what percentage that might be is very important so that you can factor that into your sample size and power calculation, essentially giving yourself a buffer to absorb some of that non-adherence and some of that loss to follow-up. They also wanted to determine how feasible it would be to actually recruit this high-risk group of older adults from diverse diverse communities and racial and ethnic subgroups within the United States. They wanted to look at, excuse me, what were the predictors of who would respond and enroll, who would adhere, and who would actually benefit from the intervention. So this is a pilot study that was actually going to run basically the same intervention as the primary trial, and they were going to try and look for predictors of who might be responsive to the intervention, who might adhere more so than others. And my guess is they were going to use those factors when they started to recruit for the larger trial. They were going to do targeted recruitment. And then lastly, they were going to try and develop a safety plan and a system to monitor adverse events that would be occurring during the course of the trial and what to do about it. And again, this is a unique trial because of who the population is. Frail, elderly, older adults. You're going to expect things to happen to these folks along the way. This is a very ambitious pilot study. But each of these points, these 7 objectives, were critical elements of knowledge that the investigators needed to determine so they could define and redefine or refine the large-scale trial so that it had optimum opportunity, if you will, to properly test the hypothesis and come up with a definitive answer to the study question. So as I mentioned, the main trial was a parallel group design. So for the pilot study, they did a small scale pilot of this parallel group design where they had a base population they recruited from. They got a study population of people who were interested, eligible, willing to be randomized, and consented, who they would then randomize to either the intervention or the control group and follow them parallel for the primary outcome. Pretty straightforward design, and I'm sure you've seen something akin to this more than once as part of our workshop. And again, this is the pilot study I'm talking about here. So as part of this pilot study, they determined that to get the information they needed at a pilot level, they would have to randomize 400 frail elderly adults into the two arms, so 200 per arm, over 9 months, and they had a targeted enrollment of 25% from racial and ethnic minority subgroups. That's what they were trying to achieve with the pilot study. So the inclusion and the exclusions are identical to what they would be for the full trial. Why? They wanted to be sure they could get the target population after they imposed the numerous exclusions that would take somebody out of the eligibility pool for this rather high-risk trial. The other thing here that I've highlighted in red as part of the pilot study, which also was part of the main trial, is that in the pilot study, the participants had to first complete a run-in phase. Now, I'm sure some of you are familiar with, maybe all of you are familiar with run-in phases. Perhaps some of you, or perhaps a number of you, have actually conducted run-in phases. Listen carefully. A run-in phase is not a pilot study. In this instance, the run-in phase is actually part of the pilot study. Typically, the run-in phase or a run-in phase is part of the main randomized trial design. And what the run-in phase is used for, it's a set of activities done prior to randomization to make sure that the person who's eligible, willing, and consented will do what they're asked to do before you randomize them into groups, and now you have to analyze them. Remember what Dr. Miller said about intention to treat. Once analyzed, always, or once randomized, always analyze. So the run-in phase is, in essence, trying to help you find people who give off a signal of being non-compliers. And at that point, you can still politely decline them from the trial without using one of your randomizations on someone who might not give you the data you want down the road. That's not a pilot study. That's a functional part of the trial design that helps weed out non-compliers. Okay. The run-in phase in the pilot study here was going to be 1 week prior to the planned randomization. So once a person made it to the study population, they're eligible, willing, and consented, they would have to do the following successfully before they would be randomized. They had to do a dietary fruit and vegetable intake record, a daily physical activity record, and daily body weights. The reason they did this for the run-in phase is both arms, the control arm and the exercise arm, were going to fill out information along these lines during the trial itself. So it kind of made sense in both arms to see, are you willing to do these 3 things as part of a run-in phase? Successful completion was designated to be 6 out of 7 days where they had complete records. If they did 6 out of 7 days, then they were moved to the randomization stage and randomized to group. If they did less than 6 of 7 days, they were told, sorry, this isn't for you, and we respectfully decline your enrollment. And they'd go out and try and find another person. Again, the whole idea here is to try and find the really low-hanging fruit. If they can't do this for a week, what makes us think they're going to do a trial intervention for an average of 2.5 to 3 years? Okay, so we're weeding out the non-compliers. As part of the pilot study, and again, this comes from the pilot design paper, the protocol was exactly the same as it was going to be in the main trial. It was just condensed into 1 year. But the people had to have everything done to them in the same manner in a clinical setting at baseline, 6 months, and again at 12 months. The difference being for the randomized trial, this would extend over 2.5 years. Whereas for the pilot study, it was 1 year. But it's all the same measurements. They're trying to tweak their protocols. They're looking for safety issues. They're looking for participant feedback on what they did. Were the questionnaires legible? Was the font size big enough? Did you understand what we were asking? And so on and so forth. Very, very involved pilot study. The other really important thing for this and many pilot studies that are done prior to the actual randomized controlled trial is to start thinking about safety. So participant safety and data integrity. And as I mentioned on the first workshop a little bit, and I'm pretty sure Dr. Miller mentioned when he was with us or with you guys, the Data Safety and Monitoring Board, or the DSMB, is the designated group of individuals who are responsible for monitoring the participant safety and evaluating the integrity of the data. So knowing that that was going to be necessary as part of the primary trial, for the pilot study, the investigators formed a DSMB. They monitored interim data reports. They looked at adverse event reports. They came up with a plan for re-entering the pilot study for somebody who had to stop for a short period of time because a health issue came up. They caught a cold, or they sprained their ankle, or whatever it may be. And then this group worked together with the pilot study steering committee to ensure that all the policies and the protocols were followed to the T, to the letter. The other thing that the pilot study, of course, was in place for, and I've mentioned this a few times, and I'm intentionally trying to harp on this, the major objective of the pilot study was to come up with a projected sample size. How many people do we have to randomize in the main trial to have sufficient statistical power to test our hypothesis at the designated alpha level for the expected effect size? So this is where they came up with that information. So they looked at the 1-year rate of the outcome, becoming losing functional independence or dying. Now notice that's coming from the control group in the pilot study. Why is that? In the pilot, just like in the primary trial, there's 2 arms. There's the intervention arm and there's the control arm. The control arm should give us the rate or probability of the outcome we would expect in the absence of the intervention. And because this is a randomized design, all of the extraneous factors that might be competing explanations for the probability of developing the event have been balanced between groups. So the event we see in the control group is the background rate or probability we expect in the community in the unintervened setting. The rate or probability we see in the intervention group, we would hypothesize is going to be lower than the control group. So the rate in the control group gives us kind of that expected rate in the absence of the intervention. And it's this rate that we use to set the effect size. The difference in probability or rate of outcome between control and intervention group, or the C minus I, as we often designate it. The other thing from a statistical standpoint that's very important, the pilot study was able to give some insight in, once you randomize someone to the intervention group, how many people drop out of it? It was too difficult, it hurt, it was too time consuming. And likewise, when you randomize to the control group, how many people dropped out of that? That amount of dropout is very important, because if you remember, the intention to treat principle is that you analyze the data based on the original randomization. However, after doing the intention to treat analysis, it isn't uncommon to then, as part of a secondary data analysis, go back into the data and reanalyze it, looking at the people who were adherent versus those who weren't. And that's where the amount of dropouts and come into play. So having some sense of what that might be ahead of time is very important when you're planning sample size. You can plan a little bit of a buffer to kind of consume some of that so it doesn't affect your primary test as much. And then the last statistical issue that they wanted to make a determination on is, knowing that these were frail elderly people, at some point, the participant themselves may not be able to fill out forms, answer questions on the phone. So they wanted to see how accurate proxy reports would be. So they got proxy information by somebody designated by the participant at randomization, who would be giving the information the study needed on the outcome. And then the investigators pulled medical records and other source criterion information and compared the proxy report against that to see how accurate the proxies were. Again, very important, because if the proportion of outcomes by proxy report ended up being higher than they expected, then they would have to defend that that data is accurate and valid. So they have some data now from the pilot study to make that justification. So kind of wrapping it up, if you will, in a nutshell, the LIFE trial was actually two parts. It was the main trial and a pilot study. Look very carefully here, top panel. This is the pilot study. The main objective of it was to estimate recruitment protocols, sample size, and event rates or probabilities. Nowhere in there did I list testing a hypothesis was an objective. It wasn't. That's not what a pilot study's for. The main trial, look what the major objective is, the very first one. Provide a definitive test of the hypothesis. How can they say it's a definitive test? Well, maybe it should be better said, provide as definitive a test as possible of the primary hypothesis. And that definitiveness, if you will, I don't know if that's a word or not, is anchored to the pilot study. Did we understand the feasibility of recruitment and optimize it? Did we have a good understanding of how many people we needed to randomize to ensure sufficient statistical power for our expected effect size at a specified alpha level? Did we have an understanding of what the safety issues would be and what the amount of dropout would be due to safety or other issues? And did we buffer against that so that we didn't compromise the hypothesis test? All of that feed into what's definitive or not. And all of that information came from the pilot study. So that at the end of the day, the LIFE trial actually randomized more than 1,600 people. It was 1,635. They determined from the pilot study that they'd need to do this over 8 field centers rather than the 3 that they used in the pilot study, so that they could get their recruitment and randomization done soon enough to still have time for the expected follow-up, which was 2.6 years. From the pilot study, they were able to determine an expected loss to follow-up of 4% per year. That was a little less than what they were thinking the pilot study would show. It was about half of what they were thinking the pilot study would show. So that worked out good for them. The pilot study suggested there would be less loss per year. They came up with what the expected events rates would be between the intervention and the control group, and therefore the effect size, and the actual effect size was a 30% probability of the outcome in the exercise group compared to a 36% in the control group, and that carried P of 0.03, which is a statistically significant difference in outcome between the intervention group and the control. And that's exactly what the trial showed. So I hope that example gives you a sense of what a pilot study is and what it's not. How a pilot study informs development and refinement and execution of a larger scale primary randomized controlled trial. And I hope that for those of you who are thinking to apply to our CTSA pilot study funding program, don't send us the full trial or the full study proposal, including your hypothesis test and statistical plan. If you can do that with pilot study funding, you don't need to do a pilot study. Conduct the research and publish it. Okay, that is not what a pilot study is for. The last thing I want to say in closing is, in spite of my intended points of emphasis, there is debate around should every clinical trial have had a pilot study preceding it? Are they valuable? And then there's even some debate around if you do a pilot study to support developing a larger scale trial, do you have an obligation to publish the pilot study findings, which is what the LIFE trial people did. I'm not here to tell you which side is correct, which is incorrect. I think it's always important that we recognize there's two sides to the coin. And we at the BERD are not here to tell you what to do. We're here to provide you advice, guidance on things that are tangible, statistical test, estimating power, sample size, choosing one design over another. But ultimately, you and the investigator have to shoulder the responsibility, and you have to shoulder the consequences of the choices you make. And I think together, we have a pretty darn good team here at UB, where we can provide you some assistance. A lot of times, one of the statisticians from the BERD find their way into a person's grant proposal to kind of anchor the statistical aspects of it. And I hope many of you, if not all of you, know, in today's funding climate, whether it's NSF or NIH, if you're looking to try and score a large scale, $400 million per year study, whether it's a randomized trial or not, and you don't have a card-carrying statistician on your grant proposal, you're opening yourself up to a ding that you don't need to take if you had a statistician there. It's just too low-hanging a fruit for someone to pick on. That's kind of the climate we're in now. So words of wisdom for the weary. I am going to stop, and I'm going to unshare so I can see everybody. I hope my screen is still there. I haven't seen much, so let me stop sharing, and hopefully everybody's there yawning and ready to wake up.